When I stumble upon single-celled organisms reproducing under the microscope, I might feel a bit like an accidental voyeur peering into nature's most intimate moments. But when I discover mass reproduction happening everywhere I look, well, let's just say I've wandered into a microscopic soap opera. But before you start blushing, let me explain what's really happening here and why it's absolutely fascinating from a biological perspective. What you're seeing is something that even seasoned microscope observers rarely encounter, a mass conjugation event. And to understand why this is so remarkable and what might have triggered it, we first need to meet the players who are about to make some very interesting moves. This is where our story begins, a small fishing pond in Bangkok. Every few weeks, I give the owner about $3 and I take home 20 liters of pond water and a pail full of floating plants. It's on the roots of these plants that the real action is happening. Meet the Stentor, literally one of the giants of the single-celled world. These organisms can grow up to 2 millimeters long. That's really big for a cell, making them among the largest single cells you can actually see with the naked eye. Named after Stentor from Greek mythology, the herald with the powerful voice. These trumpet-shaped protists are covered in thousands of tiny hair-like structures called cilia that beat in coordinated waves to create feeding currents and propel them through the water. They anchor themselves to plant roots and debris and create miniature whirlpools to sweep in bacteria and other microscopic meals. Very often, I'll see a lot of them on the roots of these floating plants. And when it comes to reproduction, Stentors usually stick to the same reliable move over and over again: simple cell division. Most single-celled organisms use the same basic move when they want to reproduce. They simply split in half, creating two identical copies. But Stentors, they have their own unique style. The Stentor divides along its length. The front half keeps the original trumpet-shaped feeding apparatus, while the back half grows a brand new one. What you're seeing here might look like one Stentor is emerging from inside another, but it's really a Stentor dividing into two brand new daughter cells. This process is called mitosis, or binary fission. Think of it as making photocopies of yourself, except you're the copier, the paper, and the copy, all at the same time. But sometimes, Stentors decide to use a completely different move. Instead of dividing, two cells come together to exchange genetic material. No new cells are created, but genetic diversity increases, which is critical for the long-term viability of the species. This is conjugation. Think of mitosis as quantity with style, and conjugation as quality through partnership. And that brings us to our mystery. Conjugation is usually rare in Stentors, at least rarely seen. So what caused this mass event we're witnessing today? To understand what might have caused this coordinated sexual reproduction event, we need to look at what researchers have discovered about conjugation triggers in ciliates. Conjugation in ciliates is often induced by lack of food. When nutrients become scarce, these organisms switch from their usual growth mode to a genetic exchange mode as a strategy to improve resilience by shuffling genes before conditions worsen. But here's where it gets interesting with Stentors specifically. Research on Stentor caeruleus has found that conjugation occurred most frequently in cultures that had been recently fed, with up to 50% of the population involved in conjugation at one time. So it may not be simple starvation. It might be the transition from well-fed to the beginning of nutrient depletion that triggers this behavior. Environmental stress can also flip this switch. Changes in temperature, pH, water chemistry, or other factors might prompt mass conjugation as a way to increase genetic diversity before conditions potentially worsen. Population density could be another factor. When too many Stentors are competing for the same resources in the same space, Coordinated conjugation might be their way of preparing for tougher times ahead. And then there's chemical communication. Some other ciliates are known to release molecular signals that can trigger conjugation, though we don't know if Stentors use this same mechanism. If they do, these chemical messages could be building up in our sample, telling everyone it's time to get busy. So we have multiple suspects, but which one, or which combination, caused this remarkable mass conjugation event? Our best clues come from research on Stentor caeruleus, and while our Stentors aren't from that particular species, what we've learned from it could still offer valuable insights. That brings us to the scene of the crime. Now let's examine the evidence. 3 weeks before this footage was taken, I collected samples from this exact same spot. There were some Stentors present, but nothing like what you're seeing now, and definitely no mass conjugation happening. So what changed? Well, timing is everything. This sample was collected at the very beginning of Bangkok's rainy season. Research has shown that many species of Stentor can undergo population blooms where up to 90% of all ciliate biomass in the ecosystem belongs to the blooming Stentor species. And these blooms have been found to correlate with increased rainfall. Here's my theory. The recent rains brought a massive influx of nutrients into this pond. Organic material washing in from the surrounding vegetation, new bacterial populations flourishing, and chemical changes throughout the water column. Increased rainfall can also provide vitamin B12 producing bacteria into the environment. Vitamin B12 is essential for many single-celled organisms. Its presence could act like a growth signal or even a mating signal, and in aquatic ecosystems, a sudden surge in this vital compound could be like flipping a reproductive switch. The sequence of events tells a fascinating story. First came the rains, then came the feast, and now the festival of genes. It appears the Stentors initially responded to these new conditions with rapid asexual reproduction, explaining the dramatic increase in numbers. And now they're switching to conjugation as that initial nutrient pulse is being depleted. This pattern might be a perfectly adapted survival strategy. By increasing genetic diversity through conjugation, just as environmental conditions are changing dramatically, they're preparing for all the fluctuations that typically occur throughout a tropical rainy season. So what does all this mean? Well, beyond just being fascinating to watch, this kind of coordinated behavior in single-celled organisms shows us just how complex and responsive these so-called simple life forms really are. Stentors play important roles in freshwater food webs. They're both predators of bacteria and prey for larger organisms. Understanding what triggers their reproductive behaviors helps us better understand how these microscopic ecosystems respond to environmental changes. Now, I have to be completely honest with you. We don't know the exact reason for this mass conjugation event. It could have been the nutrient influx from the rainy season. It could have been population density reaching a critical threshold. It could even have been chemical signaling between the cells. All I know for certain is that this was filmed on a Friday night. And here in Thailand, things like this are known to happen on Friday nights. But in all seriousness, this kind of coordinated behavior in single-celled organisms shows us just how much we still have to learn about the microscopic world living all around us. From quantity with style to quality through partnership, the microcosmos still has moves we've barely begun to understand. So keep observing your local microorganisms, whether it's pond water, puddles, or even that forgotten glass of water on your desk. You never know what fascinating behaviors you might discover. And if you do find something interesting, I'd love to hear about it down in the comments. Thanks for joining us on today's look inside the microcosmos. We hope you'll consider subscribing to our channel so you won't miss our next adventure.